Okay, hi everyone. Thank you for the introduction and thanks for having us. My name is Joe Luna and I'm here with Ricardo-Lax, and together we'll be talking about the experience of making SARS-CoV-2 replicons in the Rice lab. I'll describe the basic setup and replicon activity before handing it over to Ina, who will illustrate some really great single cycle infection strategies for replicon launch. Okay, so let's get started. Oh, it doesn't seem to, there we go. So as for many here, our work with SARS-CoV-2 started in early March of 2020, where we set out to apply our flavivirus and alphavirus replicon expertise to this new virus, linearly depicted here. The SARS-CoV-2 genome consists of two large open reading frames that are processed to form the viral replicase, while structural proteins, depicted here in black, as well as accessory proteins, are made as subgenomic RNAs. Our approach to make a replicon was to replace the open reading frame for Spike with a reporter gene cassette. We figured we would remove the key structural determinant for viral entry, its incision formation in cells. Now, one thing to point out here is that there are many ways to achieve this goal. Our spike deficient approach is distinct from very recent versions that either lack the ORF3 and E proteins or that lack the N protein. But in principle, the result is similar. This will be important when we consider potential applications for transcomplementation. More on that later. The key element for a replicon here is removing an essential structural component, thereby ensuring that no infection variants can be made. This is its primary advantage, and replicons of multiple plus-strand RNA virus families have a long history of use at BSL-2. Combined with flexible and selectable reporter readouts, replicons can serve as efficient platforms for drug development, as was done with for hepatitis C virus, for instance. In our case, we and others listed here were clearly inspired by foundational work with SARS-CoV replicons. So here is our overall replicon design. We've been using a yeast-based reverse genetic system reported last year from Volker Thiel's group, which breaks up the SARS-CoV-2 genome into multiple overlapping PCR fragments. The first 8 fragments are the same as in the Thiel lab's report, while fragments 9 through 13 we re-engineered such that one fragment would contain the reporter gene cassette in place of Spike. This cassette has a selectable marker neomycin, followed by a self-cleaving sequence, and then either a nuclear localized neon green fluorescent protein or a secreted Gaussian luciferase. So we have 2 versions. Also depicted here are 2 key mutations that we'll be using in the viral polymerase and in the NSP1 protein. We can take these 13 fragments and use them to transform yeast, which can then use their recombination machinery to assemble them into a plasmid, a bit like a large Gibson assembly. This process is pretty efficient in our hands. And here I'm showing you multiplex PCR to get a sense of plasmid assembly. And for the 6 replicons here, we get at least 2 correctly assembled clones for each one, which we can then sequence confirm. But while assembly may be straightforward, harvesting plasmid from yeast in enough quantity to make RNA turned out to be tricky. We isolated DNA and included a DNA step that would remove any non-circular DNA contaminants. We also took these yeast-assembled plasmids and propagated them in bacteria. Yields for all of these processes are typically pretty low. These are what the products look like on a gel. So here, what we're digesting with is a restriction enzyme to see if we get a distinct banding pattern that would indicate full-length replicon DNA. And as you can see, DNA from yeast is fairly contaminated. While propagating in bacteria cleans up things a bit. I should note, however, that we encounter many more mutations in bacterial clones, so this extra step can get quite cumbersome and likely reflects the bacterial toxicity of select SARS-CoV-2 regions. But if we take this DNA, do an in vitro transcription reaction, and electroporate cells, here's what we get. We get at most a little under 1% of cells as replicon positive by the reporter, and that's mainly from plasmid propagated in bacteria. So it works, but it's not very efficient. So then we next thought that because yields are so poor, and because we think that contaminating yeast DNA is an issue, why not try to specifically amplify the circular plasmid as well? To do this, we turn to the DNA polymerase from phi-29 bacteriophage. This is commonly used for whole genome amplification studies as an isothermal rolling circle amplification method with high processivity. We figured this would boost yields for our transcription reactions. Turning back to the DNA, this seems to work. In all phi-29 amplified samples, either from yeast or bacteria, we get the expected banding pattern for full-length replicon DNA. And taking this DNA and making RNA and introducing that into cells, we get a much higher proportion of replicon-positive cells. So combining yeast, plasmid saved DNase treatment, and then Phi29 amplification gave us the best results, with consistently around 10 to 20% of cells positive for the replicon. Okay, so now that we had positive cells, we wanted to see how the replicon behaves over time. Here are results from HUH 7.5 cells electroporated with a replicon that secretes Gaussian luciferase in the supernatant. Measuring luciferase activity over time, we get reporter signal that persists for a few days and importantly is about 20-fold reduced if we add remdesivir, an inhibitor of the viral polymerase. Further, reporter activity is dependent on the viral polymerase as we do not see any activity if we use a polymerase mutant. And just to check replicon activity via another means, we also measured subgenomic RNA production for the N protein. We detect robust production of this subgenomic RNA that is about 20-fold reduced when we add remdesivir, which is consistent with the reporter. And again, a polymerase deficient mutant does not produce any subgenomic RNA. Okay, so now that the replicon is working, we wanted to see how it would do across a select subset of antiviral compounds. We first tested remdesivir across a range of concentrations and measured inhibitory concentrations in the nanomolar range without seeing any cell toxicity, and this is consistent with reports using virus. We also tested masitinib, a putative inhibitor of the 3CL protease, and observed inhibition. However, we encountered cytotoxicity at higher concentrations. As a control, we tested 2,7-hydroxycholesterol, thought to work against SARS-CoV-2 at the level of entry, and as expected, we did not observe appreciable inhibition of the replicon. So this overall gives us confidence that the replicon is useful for reporting on intracellular replication apart from entry and virion production. The replicon was also useful for deciphering host factor requirements, with a special emphasis on intracellular host factors. Our lab recently identified TMEM41B as a coronavirus host factor, and we observed severely attenuated replicon launch in TMEM41B knockout cells that could be further inhibited when we add remdesivir. By comparison, a Synvis virus replicon was not affected in these knockout cells. If we use knockout cells reconstituted with TMEM41B, we can rescue replicon activity by around a log. So overall, this provides us a useful tool for host factor validation and for screening. We also wanted to get to see how replicon could be used to characterize viral variants. And here we focused on the first viral gene product, NSP1, because of its role in affecting translation and encountering the innate immune response. Using an NSP1 mutant unable to engage the ribosome, we observed that it behaves similar to wild-type replicons, could be monitored for a number of days, and could be readily inhibited by remdesivir. However, we also noticed that NSP1 replicons displayed much reduced cytotoxicity, which was a bit of a surprise. This is not to say that cells can harbor persistent NSP1 replicons, only that NSP1 from electroporated RNA appears to be a major player in inducing cell death, likely by affecting translation. Note that wild-type replicons launched in the presence of remdesivir also exhibit a similar phenotype. This further got us thinking about NSP1 and specifically how NSP1's impact on translation might affect replicon sensitivity to translation-heavy processes, such as the response to interferons. So next, we tested replicon activity across a range of interferon alpha and beta concentrations and discovered that while wild-type replicons seem to be quite resistant to interferons, NSP1 mutant replicons are much more susceptible to the action of this cytokine. This is in contrast to remdesivir, as both wild-type and NSP1 mutant replicons are similarly susceptible to this antiviral compound. Now, these points will be important in the second part of the talk, and in general, they highlight a small fraction of what's possible with this type of system. But just to summarize so far, we've used yeast assembly combined with Phi29 plasmid amplification to achieve routine SARS-CoV-2 replicon launch. These replicons can be used for everything one would want a replicon for, whether it's compound testing, host factor screening, and for doing SARS-CoV-2 genetics. And lastly here, we've observed that NSP1 appears to be a major cytotoxic factor, as well as a prime antagonist for the interferon response. However, this all points to a chief challenge of working with these replicons. Namely, RNA launch via electroporation is highly artificial, in that we are introducing an enormous amount of RNA that may trigger toxicity via RNA sensing. As a consequence, this limits the cell types within which replicons can be launched. I'll now turn it over to Ina, who will describe how we've gone about overcoming this challenge. So Ina, take it away.
Okay, so Hi, everyone. So now I'm going to tell you a little bit about how we overcame the challenges that Joe mentioned. Um, so what we decided to do is to transpackage these replicons by providing the glycoprotein or spike, in this case, in trans. And this trans-complementation system has many advantages. So for one, it really solves this like harsh delivery by electroporation problem. And also it ensures really delivery of RNA that's mostly full length, that only the RNA that can be packaged. As I will show you in a minute, when you trans-complement with Spike, it gives us an isogenic platform for rapidly generating virions that allow testing for alternative Spike variants. And then alternatively, if we provide a different glycoprotein in trans, in this case, we use the G protein of the vesicular stomatitis virus, the VSVG, we can now target cells that don't necessarily express ACE2. And then in all the things that we did, the key consideration was really making sure that the delivery of these replicons is really single cycle infectious. So the way we make transpackaged replicons, or we call them TPRs in short, is that we pre-transfect cells with a spike-expressing plasmid, and then the next day we electroporate cells with the replicon RNA. These producer cells will make transpackaged replicons, which will then we collect from the supernatant and put on naive recipient cells. So these recipient cells do not express spike, and therefore they will not make new transpackaged replicons. And if we take this supernatant and put it on naive cells, we don't expect to get any signal. To really lower any chance of the spike somehow finding its way back into the genome, we're using a spike that is essentially as in any other mRNA in the cell. So it's human codon optimized, and it really lacks any sequence that may allow, like the generally used for coronaviruses for template switching. So it doesn't have any of the 5' UTR, leader, TRS, or 3' UTR sequences. So here's how it looks. What you see here is our nuclear localized neon green. This is our reporter. And we also in parallel will stain for N protein, which you see in magenta. So these P0 cells, these are the cells that are electroporated with the replicon RNA. And you see that regardless of spike expression, we get a signal. And here we used both the wild type replicon and the NSP1 mutant replicon. And one thought was that because NSP1 mutant replicon is much less toxic to the cell, the cells are much more viable, we thought that this will help cells make more TPRs. And here, if you look at P1, these are the recipient cells, and here we used H8 7.5 cells that overexpress ACE and TMPRSS. And you see that only in the presence of spike do we get a signal in those recipient cells. And as we suspected, the NSP1 mutant replicon are more efficiently packaged, just because those producer cells are much more viable. And very importantly, these P2 cells, so we took the supernatant from P1, put them on the P2, and we see no signal, which is as expected, and we're really happy to see that. So another thing that I want to show you, and this kind of links to the first talk that we heard today, is that if you infect it with virus, you really get this syncytia formation that you heard about. But if we're using transpackaged replicons, these recipient cells, they don't express spike. And so you really get those single cells, and you don't get syncytia formation, which is another advantage of using these transpackaged replicons. So next, we wanted to see whether spike transpackaged replicons could be used for testing entry steps. And in this case, we used it for testing neutralizing antibodies. So we tested 2 previously characterized monoclonal antibodies that we received from Michelle Nuzensweig lab here at Rockefeller. So these are the C144 and C135 monoclonal antibodies against spike. And we used 2 variants of spike. So here you see the wild type spike or the Wuhan strain. And you will see that both neutralizing antibodies neutralize the transpackaged replicons really well. However, if we use this spike, and this has all the, this is a variant, this is a South African variant spike. And you see that it's neutralizing very well by the C135 neutralizing antibody, but it's really not neutralized at all by the C144. And this is consistent with what people have seen so far with pseudotype assays with lenti and replifiers neutralization assays. And then this was done with the Gaussian luciferase replicons. And here, if we compare just how these antibodies neutralize actual virus, and in this case, in both cases, was stained for N just to have a comparable readout. What you can really appreciate is that those neutralization curves between virus and TPRs, they look really pretty identical. And so this shows us that TPRs can be really reliably used for these types of assays. And the big advantage of using TPRs actually over even using the actual virus is that you can really easily swap different spikes and test different spike variants, which is much harder to do with virus. You have to either engineer a whole virus clone, or you have to wait to get some isolates. So this is a much faster way. Okay, so now we have a system that we can use to study intracellular viral activities as well as entry. But I guess the main limitation is that you need still ACE2 overexpressing cells to get efficient entry for spike TPRs. So that significantly limits the type of target cells that we can use. So the next thing we decided to do, to borrow a page from the lentivirus book. And we used the VSVG express and trans to package the replicon instead of using spike. And this is again very similar to what people are using for lentivirus particle packaging. And with lentiviruses, we know that we have a single cycle infection or transduction. And here, very similar to spike, we pre-transfect cells with VSVG. We then later electroporate with the replicon RNA. And these producer cells will make TPRs, which we'll use to inoculate recipient cells. And this will be a single cycle. So this is just VSVG instead of spike. So here, very similar to what we see with spike, we see a signal in the producer cells regardless of VSVG expression. But in the P1, we only see signal when we have VSVG. And again, with the NSP1 mutant replicon, we get much more, we get many more TPRs. And again, similarly to spike, passage 2 shows no signal, showing that it's really single cycle replicons. So because this point was very important to us, we also repeated this for the more sensitive readout, where we used Gaussian luciferase replicons, which we then further concentrated. So the idea is this: we take TPRs and we infect cells with the Gaussian luciferase TPRs in a 6-well format. After 1 hour, we take this initial inoculum and we concentrate it by PEG. And we test this, the presence of TPR in the supernatant by infecting naive cells and measuring the Gaussian luciferase signal. And you can see it here. We get a nice signal, which means the inoculum is there. The PEG precipitation works. But then we take these cells, we incubate them for 24 hours. We get the replicon, replicating and expressing the reporter. When we measure luciferase from the supernatant, this is the P1, we get a really, really nice signal. But then if we concentrate the TPRs, or the potential TPRs that are in the supernatant and infect naive cells, there is really nothing there. So this really demonstrates that the system is clean. It's really a single cycle system. So, so now we can use these VSVG TPRs for its intended target, which is basically infecting different cell types. So here on top, you see cells that are, I guess, normally used for SARS-CoV-2 research, HLA 1.5s, Vero, Keiko2, and Calo3 cells. But here underneath, here, for example, the A549 cells, we know that we cannot infect them with SARS-CoV-2 unless we engineer them to overexpress ACE2. But they are susceptible to the VSVG transpackaged replicons. These are primary cells. These are normal human bronchioepithelial cells and lung fibroblasts, which are really much harder to engineer than a cell line. And we can see that they are too susceptible to the VSVG TPRs. And we can use these cells now to pre-treat them with drugs. So here, for example, we use remdesivir and interferon alpha pre-treatment, and we see that it nicely inhibits the Gaussian luciferase signal from the replicon. So now potentially this can be now used in high throughput screening, and also as opposed to working with electroporation, we're sure that it can be used in a more complex system. For example, lung organoids or different systems. So next, what we wanted to do, we wanted to add additional layers of attenuation to the trans-packaged replicons. So one of these strategies was really to work with the NSP1 trans-packaged replicons, because we already showed that NSP1 is really important for reducing the interferon response. So potentially these might be less pathogenic in vivo, although this still has to be tested. But the other strategy was to really remove all the accessory genes. And we know that the accessory genes are not strictly required for replication, but they do play an important role in pathogenesis. So we removed them and we only left the structural genes, so E, M, and N. And what we nicely see here is that these delta accessory genes, replicons, can be also trans-packaged pretty efficiently. And this is, of course, VSVG dependent. So we can trans-package them with VSVG. And this is great because first we added another layer of attenuation, but also now we can start testing the activity of these different accessory genes, either alone or in combination, in the context of virus replication. So just to summarize the second part, the trans-packaged replicons allow for authentic single-cycle infection for replicon delivery. The spike trans-packaged replicons, they really represent an isogenic platform for testing different spike variants. And then we can use that, as I showed you, for neutralization assays and other assays that have to do with entry. Pseudotyping with VSVG really allow for flexible replicon delivery to different culture systems and different cells. And then lastly, I showed you that the minimal replicons that lack all the accessory genes can still be packaged. So the last slide, I'm going to show you things that didn't work, and for one we're still working on, but the other one we're really happy it doesn't work. So first of all, things that we're still working on. So Joe mentioned that in addition to the reporter, we'll also have neomycin selection gene. So we've been trying for months now to select for stable cells that harbor persistent replicons. And we weren't successful until now because the cells just died eventually. So I guess the replicons just was too cytotoxic, even though we tried also using the NSP1 mutant.
Ina, I'm
yeah.
sorry, this is a 2-minute warning.
Yes, I'm almost done, thank you. And so now what we wanted to do is try doing that in different cell lines, because maybe we can achieve stable expression in other cell types. And this is where the VSVG trans-packaged replicons come into play, because now we can target all those different cells. The second thing that we were not successful in doing, and this is really good because we're trying to kind of see whether we can get infectious virus from any experiment with trans-packaged replicons, and we didn't see any of those in any of the experiments that we did. And we think that partially is because we deliberately took out all these coronavirus sequences that can mediate template switching. But again, we have a secondary line of attenuation by NSP1 mutation and accessory gene deletion. So with that, I want to thank you for your attention. A special thanks to Charlie Rice, our PI, who's been really hands-on during all this process, and it was really an incredible experience to work with him. The entire COVID team in the Rice lab, our collaborators in University of Bern, Volker T lab for help with the yeast recombination, Rockefeller labs, Route lab for help with yeast work, Binash lab for spike variants, Nusen spike lab for neutralizing antibodies, and other collaborators, JT Poirier for NOU and Francisco from the Scott lab in MSKCC. thanks to our Rockefeller Biosafety Committee and the Rice Lab admin and support staff. And of course, this work was made possible for our funding, Matters Foundation, FAST grants, NIH, and my fellowship was Bulgari Women in Science Fellowship. Thank you.
Thank you very much, Ina and Joe. That was great. I remind attendees and panelists too, if you have questions, please raise your hand and we will unmute you so that you can ask your questions. I'd like to start off with, I guess, a practical question. In your system, how long does it take to generate, for example, a particular mutant that you would like to assess? And does that time scale linearly with the number of mutants that you would like to test? So for example, let's say you had 100 mutants that you wanted to do. How much time would this take?
Yeah, I guess I can answer that. It takes, you know, including making sure that everything's in place and sequencing what comes out. It takes about 2 to 3 weeks. But the idea is that the system is really modular. So if you want to introduce a mutation in a specific gene, all you need to do is replace this fragment. You don't need to, you know, to clone the whole thing. But you will have to do another recombination in yeast, kind of to get everything in place.
Yeah, I would also add that the limiting factor seems to be, yeah, like sequencing. You know, yeah, sequencing, making sure that you know, have the clone that you want. Because once you have that, to make, you know, to 529 amplified and make RNA is pretty fast. That's like within a day.
Perfect, thank you. We have a question from Steve Goff. Steve, go ahead.
Yeah, hi Joe, Ina.
Hi.
So one of the nice implications of the particle production is that, I guess, just like GAG-Paul, you know, for lentiviruses, your replicon is making particles, you know, nicely, even without any envelope, without any spike or VSVG. So I wonder, have you looked at the particles? I mean, are they looking normal, except they would be bald particles, presumably, right? Without the hair of any envelope.
Yeah, that's what we're planning to do now. Like we were, yeah, like the answer is no, we haven't. But I mean, this is on the docket to really see, you know. And one implication there is that, yeah, it might explain the relative inefficiency because there might not be enough spike in a lot of these baldish particles. And so, you know, like we were thinking how this might be a way, if we could titrate the amount of spike that's on a particle, we might be able to get at some questions as to how much do you need to actually cause an infection.
I mean, yeah, you could imagine that having an envelope of either sort would stimulate particle production. I mean, you don't know. GAG-Paul doesn't care, actually, whether you do or don't have an envelope. It makes almost the same yield of particles either way, but that might not always be the case. And you could imagine that a good envelope, either VSVG or its spike, is helping particle production per se. So, you know, maybe just a Western of the particles, you know, would tell
Yeah.
you.
Mm-hmm.
Yeah, it's a really good idea.